Okay, so this is a kind of recapture of our journal club because we had some IT issues where I kept being bounced out. So just want to kind of create a more seamless discussion about the journal club. All right, so in the journal club paper, we kind of a couple different players involved. And so we have COVID-19, so our viral infection that affects a number of organ systems, but the respiratory organ system being probably the one that's most prominent, if we will say. And then the other players are C5a, and so you'll recognize C5a from the complement system, hence why we picked this paper for this journal club. And so we have the complement system being involved. You'll remember from our lecture that C5 will cleave and it's going to form C5a and C5b. C5b goes on to form the stalk of the MAC, the membrane attack complex. So to form that membrane attack complex, we're going to have C5b, C6, C7, C8, and C9s. And we can have anywhere from 6 C9s to 20 C9s forming that MAC. This paper, we're not going to care about C5b. It gets enough press. What we want to focus on is C5a. And so this can go off and cause inflammation. And so it also can bind to C5a receptor. Okay, and so macrophages, neutrophils, a number of innate cells will have this receptor that can bind C5a. And when it binds C5a, it's going to activate. So very much like toll-like receptors, very much like pattern recognition receptor, these complement receptors, when they bind their ligand, become activated and signal to the immune cell, we saw what we're supposed to see. Now we need to respond. We need to do something about it. And so it'll cause cell signaling events that will cause the cell to produce cytokines, to potentially proliferate and such, and activate other cells. So that's a little bit about the players. And the figures are at the end, towards the end. So you just scroll down. Now in this first figure, they are looking at COVID patients. And so they are looking at blood samples. So they have blood samples and they're looking at these different cytokines that are present in the blood sample. So in the serum of the blood, in the plasma. And they have 4 different groups. 3 of the groups are infected with COVID-19. So they have a mild case. They have individuals with pneumonia, and then they have individuals with acute respiratory disorder. So ARDS. And so ARDS would be the most severe. This is individuals that are going to be in ICU, intubated, need to be on supplemental oxygen in order to breathe. So their respiratory system is quite affected by the viral infection. The pneumonia individuals, they're having difficulty breathing, but they may not be to the state, they're not to the stage where they have acute respiratory distress. Now, keep in mind, pneumonia can be bacterial or viral. So pneumonia is a general term when you have an infection in the lungs that are making it difficult to breathe. Okay, and then again, we can have mild cases also. We could also have asymptomatic cases, but if you go through the materials of method and look at how they put people in the study, their age groups and all that kind of stuff, you had to have a positive PCR test. And so our healthy controls are PCR negative for COVID-19, but they've had blood samples taken. And then in a future figure, they're going to do BARF, which is a bronchial lung lavage, where they get fluid from the lungs and look at the sample. But again, this is in the blood, and they're looking at the different cytokines. And you can see that for individuals that are in, let's switch that color, that are in acute respiratory distress, they have elevated cytokine levels, except for CCL2. But for all of the other cytokines, all the other cytokines in panel A, they have elevated levels. Now, when they go and they look in panel B at C5a, so complement protein 5, but the A subunit in circulation in the blood, they see that this increases with severity of illness. So that goes up. And again, keep in mind, this is in the blood. So that these cytokines are in circulation. In our next figure, figure 2, they're looking at the cells that are present. So if we have cytokines that are increasing, if we have C5a increasing, how does that affect the cells that are present? And again, they're looking in the blood. So we do see elevated neutrophils in circulation in peripheral blood. Now, when we look at monocytes, you'll notice that monocytes, there's no statistical difference between the different groups. They also look at monocytes using some different markers. So they're looking at more specific subsets of monocytes. And so for their, you'll notice that they're the same for the first markers, where they're looking at CD14 positive, CD16 negative. When they're looking at CD14 low and CD16 positive, you'll notice that there's actually a decrease in these monocytes being present. These monocytes, CD14 low, CD16 high, are indication of inflammation. So when we have inflammation occurring, inflammatory events, these monocytes usually are increased. So this is kind of counterintuitive of what they would think was going to happen. And so one of the things they have is a big question. And their question becomes, what about in the lung? Okay, so are these inflammatory monocytes elevated in the lung, and that's why they're decreased in the blood, right? So we know with COVID-19, especially with these acute respiratory distress individuals, they're going to have a high amount of inflammation occurring in their lungs. And so is that where the monocytes are? And at this point, we don't know. It's just an area for like, oh, let's see, maybe. So they also then look at C5a receptor positive. So these are cells that have the receptor to C5a. They look at both neutrophils and monocytes. So in panel C, what we're seeing is monocytes, neutrophils, and then monocytes. And what they notice is that for all of these conditions, healthy control, mild pneumonia, and acute respiratory distress in the blood, there's no difference in the level of these cells. So again, keep in mind, this is all in the blood. And then for our last panel, panel D, what they've done in this panel is they've taken peripheral blood monocytes. So peripheral blood monocytes. They've isolated those. So taking some blood from individuals, purifying out these cells, and then they set up a couple different conditions. They add LPS. So LPS comes from gram-negative bacteria, lipid polysaccharide. We know that this activates TLR4. So they activate TLR4 with LPS, and they have that alone in a condition. And then they also test it when we add C5a. So we add this complement protein, we've activated. So kind of like our last Journal Club paper where they take those macrophages and they add ligand and like CPG, and they activate those macrophages. So we're feeding something we know that it's going to kind of piss them off a little bit, and we're saying, what's the cytokine levels? And if we add in C5a, so we even make them more angry, will this make them produce even more cytokines? And so the other thing they add, the other condition is adding R848. And so for that, that's going to activate a different TLR. And so that's TLR7/8. Okay, so that one is a heterodimer. TLR4 is a, nice, transpose that. TLR4 is a homodimer, right? And again, in the condition where they just add R848, they also have a condition where they also add in C5a. Okay. Now the reason they're doing this is so that the effect we see is not just with one, is not novel to TLR4, right? Because if you just did LPS, the argument could be, well, the only reason why you're seeing effect is because you're activating TLR4 is something unique to TLR4. What about the other TLRs? So they really could have picked any ligand to any other TLR, or maybe they did, but they could also do CPG and add in C5a and see how does that change. What they see in the conditions with C5a is that for each of these conditions, regardless of what TLR ligand they received, there was an increase in cytokine production in both cases. Okay, so you can see how those lines traject up. So again, an increase. Again, this is still all done in the blood. This is done with cells isolated from the blood and then primed. And so then in figure 3, now we're looking in the lung, okay? So what they've done is this bronchial alveoli lung lavage. And so they collect fluid out. So when people with acute respiratory distress, when they're intubated, there's going to be a buildup of fluid, and so they have to get that fluid out anyways. So for these individuals, it would be it's part of the practice of making sure that buildup of fluid is removed, that you could get those samples. Okay, so with that, they look at the different cytokines that are present. So that is what we're looking at panel A. They look at C5a, and so you see C5a upregulated. You also see CXCL8. You see additional chemokines upregulated. So again, in these in the bronchial lavage, in that fluid that's being collected from these acute respiratory distressed individuals, we are seeing cytokines being present in those. They then look at this, the cells that are collected in that fluid, both at neutrophils and monocytes. And so the red is our acute respiratory distressed individuals. And you can see that those cells have higher expression of C5a receptor. So remember, when we looked in the blood, there was no difference between the different groups and C5a receptor positive cells. But in the lung, when we see that shift to the right, there's more of them. So they weren't in the blood because they were there in the lung. They further look at characterizing the cell population that's collected, and they do have healthy controls to compare this against. And so you can see that in panel C, they are in different locations. So there's differences between the acute respiratory distress cells that are present and the healthy control cells. Furthermore, when they look at expression levels and percent positive, the top part is our acute respiratory distressed individuals. And the bottom part is subset A, which is our healthy individuals. And the size, the diameter of the circle indicates it's a yes, no. Does it have this present or not? So you can see with subset B, our acute respiratory distressed individuals, those circles tend to be larger for these different cytokines. So they have more cells that are positive for those cytokines. And then when we look at the intensity, like how much they have of it, you'll notice that many of those in our subset B have a higher expression level, where when we're looking at subset A, our healthy controls, those tend to be low level of expression. So, you know, looking at the lung, looking at fluid collected from lung, because we don't have biopsies done here, right? But this is collecting that fluid that's coming out of that cavity. We can see that this is where the cells are. So in our last figure, figure 4, now what we do is we add drug treatment. Okay, so imagine we have our cell here. It's gonna have C5a receptor on the surface. We have C5a in circulation. We know that if it binds to the receptor, we're going to get cytokine production. We're going to get these cells being present in the site of inflammation, moving to the site of inflammation. So what they're asking is, what if we block it? And let's switch that to purple. So what if we have an antibody that recognizes the receptor and blocks it, so C5a can't bind the receptor? So this C5a comes in, but the antibody's blocking it, so it can't bind its receptor. So the cell's not going to be responding because the receptor is not binding to its ligand. We don't get the cell signaling events. So we can look at, you know, downstream, you know, NF-kappa B activation, all this, these other transcript factors, but we can look also at cytokine production as like an endpoint, right? And so they have this drug, avaquim, and so this blocks it. So this is actually a drug that's designed for cancer, but because we knew that there was elevated C5a levels, we knew that this antibody, this drug blocks C5a receptors. The question is, can it be used with COVID patients? So what they do is they look in these different treatments, and where they go and they give humanized C5a, so that's going to activate, right? And then they also do conditions where they have an IC stands for isotype control. So this is a negative control. So this antibody doesn't bind to C5a, doesn't bind anything specific. And so again, it's like saline, it's like the media, it's like mock, it's a negative control. And then they add the drug, which is going to block it. And again, they use different measures of seeing whether that's actually affects it or not. So these studies in this panel are done on humanized mice. So they're mice that have human HLAs, human aspects to them. And so that's why they're using, they're not using mouse C5a, they have to use humanized C5a. So that's where you see that HU C5a, okay? So in these mice, in panel A, we're looking at the number, the amount of CD11B, which is our neutrophils that are present. And so we can see that we shift. So wherever we see the purple line is with drug treatment. We see a shift to the left. So there is going to be less neutrophils present. These cells further produce less C5a, right? So they don't activate as much C5a when you have drug treatment present. When we're looking at migrating, So neutrophils that can move to different location. Again, when we add C5a or C5a with an isotype control, we see increased migrating neutrophils. So neutrophils moving to location. When we add our drug, we see that goes down, right? So drug blocks the neutrophils from migrating. Chemotaxis, right? They're not responding. They then, in panel D, use a couple different markers. So first, they just are looking at immune cells in general. So CD45 is just a general marker. In the middle, they're looking specifically at neutrophils. So they use two markers of neutrophils. And then on the end, they're looking at monocytes. So same conditions. So media, PBS, someone didn't keep the labeling consistent. PBS is just your negative control. You add our humanized C5a, and we expand the number so that we have more of them present. Isotype control, again, more of those present compared to control. And then we add our drug that's going to block C5a from binding the receptor. And we see a decrease in the amount of immune cells, so CD45 positive cells. We see a decrease in neutrophils, and we see a decrease in monocytes. Okay, so all of those markers, all those different cell types, when we add drug, when we block C5aR, it becomes decreased. And I'm going to have to move this and position it wrong. In panel E, they measure albumin levels. So same kind of setup where we have our PBS, our negative control, we have our humanized C5a, and then we also have humanized C5a plus isotype control. And then we have our drug treatment. They're measuring albumin because this is a marker of lung injury. And so when the lung gets injured, it releases protein into the serum. And so albumin levels would go up to indicate that we have lung injury. You can see with both our C5a and C5a with isotype that this is elevated, but that level goes down when we have drug treatment. Okay, so drug treatment blocking C5a receptor leads to a decrease in albumin levels, which is indication of better, you'd have better pathology of lung, so less lung injury. Now they don't have like histology slides. They could have, it might be in supplemental data, but that piece is missing. They then in panel F, they are looking at their cytokines, so IL-6, TNF, and CCL2 to look at, again, whether this drug has an effect. So they are using R848. And so again, this is going to lead to TLR6 and 8 activation. They also add in C5a. So again, it's a, let's go over the top. They use the isotype control just to make sure it's not the mere fact of an antibody being present. And then they add their drug treatment. And so in each case, when we are blocking C5a receptor, we see a decrease in the cytokine being produced. So, which would speak to why we're not seeing the expansion of the number of cells present, why we're not seeing lung injury, right? So panel D and E are kind of before the cytokine storm, right? And so F is saying, well, the reason we're seeing the decrease is because cytokine storm didn't happen, right? So the cytokines, as shown in panel F, were not produced. All right, so hopefully that it is more seamless, and maybe helps in understanding the paper. If you do have any questions, please ask. And yeah, ask, email, if you have any questions about this. Again, big pieces here are, the idea of the cytokine storm, the role of C5a in inflammation, right, causing that cytokine storm. Also the concepts of like expansion of these cells, right? So it's not that C5a is just there, it does something, causes inflammation. It causes, because it binds the receptor, it signals to those neutrophils and those monocytes to expand, increase in number, and go to the location where inflammation is occurring. And in COVID-19 patients, that's the lung. Now, one would argue that they could look in the GI tract. We know COVID-19 affects the GI system because there are cells called tough cells, TUFT, and they express ACE2 receptors. So that's a target. We also know in the heart, COVID-19 targets the heart. And so it would, to kind of say future experiments, some of the directions this could take is looking at these other organ systems that are affected by COVID-19 and saying, does this drug have an effect? Could this drug be used? Now, this drug was already being designed for cancer therapy, so that's why it got kind of a quick turnaround and initially used. It ends up the long-term, there really isn't any kind of long-term benefit with the use of this drug. It is also extremely expensive. And so there's other treatments that seem to be more promising, like the monoclonal antibody therapy and just vaccination. So again, you're not seeing this treatment being utilized for COVID patients currently. So, all right, thank you very much.